right. Well, we have some people here. Nice to see everyone. I know most of you, and I'm really happy that you're here. I'm going to talk through this analysis tutorial of how to run a genome-wide association study with BioData Catalyst powered by Terra within data from the TopMed study. So this is really a very TopMed specific tutorial because we're going to leverage various pieces of BioData Catalyst in order to gain access to TopMed data, which requires you to have access to TopMed data, which is not everyone in the world. But as I go through this presentation, I will ask that we not interrupt during certain sections so that I can get recordings of them, of these sections, as I do them live. But I will pause and we can actually have an interactive discussion and you guys can ask questions. I realize that we probably won't get through all the steps in an hour. And I only realized this a couple minutes ago. So we will most likely stop after we've set up the workspace for the Genesis analysis. And then what I'll do is record a few more videos so that everything is made available. And if there's a desire, I'd be more than happy to meet in a couple days or next week in a very, in the same forum for us to talk through the last few steps. Because as you can see from my screen, there are, as I'm counting it, maybe 11 big steps to this analysis pipeline. And there are even in there's so much more that you can do. But I'm going to get started with the first of our analysis steps, which is obtaining phenotype data from BioData Catalyst powered by PIC-SURE. What I have to share with you is a video of me going through the steps in BioData Catalyst. I will narrate and I'll pause at the end and ask if there are any questions and we can have a short discussion. So let's get this video started. All right, so I'm starting from the BioData Catalyst website and I've clicked on the PIC-SURE service. And now I'm logging in with my ERA credentials. And because I have access to TopMed data through dbGAP, I can go into the authorized access and start searching for my phenotype. So this analysis is going to showcase blood pressure in two cohorts, the Amish cohort and the Mesa cohort. And so you can see here that I've searched for blood pressure and found it in the DCC harmonized data set. So I've done my reading and designed my analysis plan after studying the data available. I know that the study variable is going to allow me to restrict the data to only the cohorts that I'm interested in today. So I'm looking at what comes out when I type for study, and I've chosen one which is allows me to restrict to the values of Amish and Mesa. So there just happens to be some sub-studies with each of these data sets. And I have now created a sample of 9,381 individuals comprised of these three cohorts who have the diastolic blood pressure value. And now I'm going through all the available data. And I've realized, or I've set up to download both the demographic data set and the blood pressure data set from the harmonized data set available in TopMed. And I know that I also want to get a few more columns of data like the study ID or VCF sample ID, the consent that consent group that each person belongs to, and then I export the data. So after a couple seconds, there's this query ID that is available to me, and I copy it to my clipboard. And that is the end of our first demo. And here what I will point out is that I was familiar with PIC-SURE when well, actually I've become familiar with it over the last few years, and played around with a bit just to make sure I knew the steps that I needed for this particular demo. So the great thing about the PIC-SURE resource is that there's an open access view into the data variables that are available. So anyone who has an ERA Commons account can actually browse the PIC-SURE site. And I would encourage everyone to go to the documentation available in BioData Catalyst and learn more about PIC-SURE. I'll stop and ask, see if there are any questions. Oh, so we're not, this is more of a, I will say that you should know what data you're looking for. Otherwise, you might be slightly frustrated because, as you can imagine, there's just so much data available to us. So I will now go to the next portion of our analysis, which is obtaining genotypes from BioData Catalyst powered by Gen 3. So another service currently available in BioData Catalyst from which we can obtain files that are made available on dbGAP data. I will say dbGAP data sets that have been ingested into BioData Catalyst. So here I've gone to Gen 3 and gone into the area where I can explore data and data files specifically. I know that I'm going to be looking for the freeze-8 VCF files. So I've examined the data sets available to me, selected call set of freeze-8. And now I'm going to be searching for the particular project IDs that I want to obtain for use for my analysis. So I've got the MESA data from TopMed, and now I've got the Amish data in TopMed. And I can look at the files that these results return, and they're mostly VCF files, except there's this sample.txt. So I go ahead and select VCF because I only want the VCF files. And now just kind of looking around, looking at the files, but then decide these 69 files are the files that I want. So I'm going to export these to Terra. And this may take a minute, but the format of the data is very structured, and within the BioData Catalyst documentation, you can learn a lot about the structure of this data. We'll see a bit of it as we look at the workspace, but there is a very important, a very nice method to how the data is organized and made available to us in our workspace. So here I am. Finally, I've waited a couple seconds, and it has been set up to be exported to our workspace, and I'm going to create a new workspace. In this instance, I'm going to call it my BioData BD Catalyst freeze-8 rare variant GWAS tutorial demo. And I'm going to select one of my lab billing projects. And I'm going to create this workspace. Now, this is going to start an import of links to data into a Terra workspace. In just a second, we will do a tour of the workspace. You can see here, the video is just about done. Every once in a while, you see a little glitch like that, and you just work your way through it. And I think this is the end of my video. Oh no, my video keeps going. Let's see. I think I'm going to stop the video here because what I want to do is bring up the workspace for us to look at interactively. And I'll give you an introduction to Terra along the way.
Alisa, can I ask a quick question about the picture step?
Sure.
So you mentioned, you were selecting the boxes for which data sets you wanted. And you mentioned you were copying something to a clipboard. what, how are you then getting that? Are you downloading that data locally when you're checking the boxes, or what exactly is happening?
No, what it does, the data is being prepared for you to get in a subsequent step, which I will show you. You will only be able to get that data after a check on your credentials to make sure that you are, in fact, authorized to access that data set. So I will show you how we import a data set from PIC-SURE into the workspace in a notebook.
Oh, got it. Thank you so much.
Sure. PIC-SURE does have a lot of different functionalities. There is the ability to download one of these phenotype files directly to your computer if that's what you decide to do. So here we are in the workspace that I created when I made the video a little while ago. I haven't added anything to the dashboard, but when I delete all the individual level data and make a template that people can use, I will fill in this with the documentation that I have. But a workspace has these various tabs. If you click on the data tab, you'll see that there are these tables. After the first step, which I stopped the video halfway through, it would have shown 3 tables. And this reference file is one of the tables that has very well-defined structure, which represents the files that I downloaded. So you can see, for example, the file names are the same file names that we were just looking at in Gen 3. I have restricted the view of all the columns using one of the features of Terra. So there are many columns, and some of them are interesting and useful, but for the moment, I only want us to look at this column here, which is the DRS URI, DRS URI from GA4GH, the Global Alliance for Global Health, an organization that is setting some standards for open and fair access to computational resources and data for scientific research. So what this DRS URI, is a link that I don't have to think about what this link does or where it is, but it's a link to the data set, in fact, the same data set that is in this file name column, so that when I want to use this data in a workflow, I'll just provide this path, and the workflow will automatically go. When I run it, that file will be copied into the little virtual machine that is running my workflow without me having to copy it to this particular workspace and potentially have to deal with these really massive files and pay for the storage of these really massive files. Of course, if I derive files from this file that's behind this link and copy it to my workspace, I will have to manage, the cost of storing those files. But at least the original files are not you don't have to worry about a manual step of copying them to your workspace. And I will show how we're doing that in a couple steps. Okay. The next thing I'm going to show is how a person gets to be authenticated in such a way to use a file like this DRS URI, this file link in an analysis. And now what I'm going to do is show you guys dbGAP. I had my window open, so I'm just going to create a new window. No problem at all. Maybe I'll just... All right, so I'm going to go into dbGAP, and I am the PI of a dbGAP application for TopMed data. And this project has some downloaders that I've added, and these are members of my lab or collaborators within my group or within my institution who are authorized to access data via the DRS URI in a Terra workspace, and authorized to access data through PIC-SURE and Gen 3, and the other components of BioData Catalyst, like the 7 bridges analysis workspace. So as PI, I have the ability to add people to my dbGAP application. It's not the only way that people can interact with BioData Catalyst, but it is required in order to access the data through the dbGAP. And the next thing I'm going to show you is how within your Terra workspace, or within your Terra profile, you can sync your ERA Commons account in Terra. So I've gone back to Terra and selected on my name and in profile. And here we see the various identities on the right that are linked to my Terra account. And importantly, there's an NIH account. So right now I am authorized to access, but if I wanted to renew it, and you have to do this every 30 days, it tells you when it's going to expire. So there you go. I go to an NIH login page and log in with my credentials, and I will have renewed my access to BioData to NIH accounts. And I could do the exact same thing with the BioData Catalyst framework services. So this having an active link in my profile for the NHLBI BioData Catalyst framework services is what allows me to access the data through the dbGAP. I'd like to stop here and ask if anybody has any questions, because I know it's a little confusing and it took me a while to get my head around this concept. And we also obtained these files from Gen 3. So first we had to log into Gen 3, authenticate, and we would have only been able to authenticate if we had our name on the dbGAP application linked. And the data sets that I saw in Gen 3 would only be the data sets that I have access to. And then I exported them to my workspace. And now in a subsequent step, I'm going to use this file link as input to a workflow. And at that point in time, there's going to be a check to verify that that I have the ability and the permission to access this file.
A quick question, if I may.
Oh, sure, absolutely.
So all of this, like you said, these are like VCF files that you've been able to sort of identify through Gen 3. Later on, will we sort of circle back to what you did in PIC-SURE, where you got the phenotypic data? Like, will it all sort of come together? So we need both pieces. Okay, great.
Yep. So in fact, why don't we go back to my presentation and take a look at the outline, and you'll see. Oh, actually, hold on a sec. There we go. I will first finish my tour of BioData Catalyst, powered by Terra. We looked at the data tab, and importantly, this is a way and a place that data can be organized in your workspace for analysis. I'll very quickly we'll look at notebooks, and then we'll definitely come back in like maybe step 6 or 7 and dive into these notebooks. So in a notebook, you have a cloud. Well, in your workspace, you have a cloud environment, which you can use a default configuration or select other configurations. So each one of these environments has a certain set of tools installed. And I can click on what's installed in this environment. And just actually see what Python libraries are installed. I'm not much of a Python programmer, so I'll go and look at the R libraries that are installed. And don't worry, you can actually install libraries on the fly while you're running a notebook interactively. So I some of these well, the point I want to make is that our studio is available here, and that more could come if there was a desire and energy towards making other environments available. So when I want to run a cloud environment, I can start or update my environment and click this button here, which is Start Cloud Environment, which means a virtual machine with the configurations described here with only a single CPU and a small amount of memory. This machine is being spun up in the cloud and assigned to this workspace so that I can run these notebooks interactively. Workflows behave a slightly different way. So a workflow is here, I'll go to the Genesis workflow where we set up the first step of our analysis, which I might not be able to get to today, but at least we'll get a look at the workflow. So a workflow has a defined set of steps. And this is in the workflow definition language, WDL, which this whole file is the WDL file, the workflow file that defines what's going to happen. So it's static, it's not changing. This is different from an interactive analysis where I can start running an R notebook and be playing around with things. So once we get to a point where we know exactly what we're doing and we want to optimize it, we'll build a WDL. This particular WDL has several tasks that run and summarize the phenotypes for a genetic association analysis that uses the Genesis package. And I would say that it's always important to review the workflow. One of the labels in a WDL is this with a label called workflow with the name of the workflow afterwards. And each input to the workflow is defined, and the tasks that are called are listed. So there's a null model step, there's a null model report step, and there is a way that you can click on the source of the workflow, go back to the beginning. You can examine the scripts and that are used in this workflow so that you can become comfortable with what's happening. It's all very transparent and reproducible and versioned. So it's a nice set of tools in order to be able to reproduce, but also debug and extend the analyses that you want to do. And speaking of debugging, I have here a couple examples of workflows that are running or that have not run in the workspace. And I'm going to show you how you can debug a failed run and using the features of Terra. So when we look at a particular submission, we have submission details. Nothing really ran in this case, so there's not a lot, no run cost. But if I want to try to understand what went wrong, we have this job manager, which shows you exactly what were the inputs to the workflow, down to the files, every file that is input. And in the case of this, it's using a file that's in the current workspace Google bucket. So I had created the phenotypes file that would be used in a genetic analysis. But there's many different ways of examining what went wrong. The log tells you every step of what and displays the output. So you can actually see what is called by the scripts within the task. And then, you can see it says no such file or directory. And looking at this, you can see, well, that doesn't look like potentially a correct file name. Maybe something went wrong, and you can start the debugging process. Okay, with that brief tour of a Terra workspace, I'm going to pause here and ask if there are any questions about the workspace, and then I'll get back into setting up the workspace for the analysis, dealing with phenotypes, and preparing data for the VCF data for merging, and getting it ready for analysis. I will go back here and we will look just briefly to see where we are in our analysis steps. No questions? Kind of lost track of my screen, so here we go. Is there anything in the chat? Are we good, Tiffany?
Yep, we're good.
All right, wonderful. Okay, so far I've talked through getting data for your analysis, and now we'll focus on setting up our workspace for the analysis that we're going to do. And the steps that I'm going to show you are actually very specific, because I've written notebooks and workflows and workflow configurations that all work together with this particular setup. Terra is quite nice in that you can set up your analysis in different ways. And this is only one way, but I hope that by making a template available, basically a pre-set up workspace without any data, but just set up, that this will make it easier for all of you to start using Terra for your top meta-analyses. All right, so let's go to, oops. I hit escape. And I wanted to make sure, okay, here we are. So I need to click through to my next. All right, so now I'm going to show a video of how we interact with the data model in our workspace in order to set some variables that will be useful in our analysis. So here I am in my brand new demo workspace. I go to the Data tab and select Workspace Data under Other Data. And this is an opportunity for us to set variables that would need to be accessed just kind of as a global variable. So you can see here that I'm created a key variable, a variable with the value demo. So the name of the variable is current analysis, and the value is demo. This will be used in the next step, which is where we run a notebook that will install packages and set up our analysis. So this particular this notebook will be available for anybody to run. Is not specific to any analysis. In fact, the first thing that you do in this notebook is you install this helpful bioconductor package called Anvil. And Anvil is another flavor of Terra that is being used by NHGRI projects like BioData Catalyst powered by Terra. So I am now using these libraries to interact with the data in the data model. And you'll see that when this block of code completes, it'll print out a table and a variable analysis label, which is the variable that I just set in the workspace. Yep. So in this data table, there is a variable demo that I'm going to use later on. So the AV table function prints out, allows you to access tables in your data model. So I just looked at the projects table, and now I'm looking at the reference files, which, if you recall, was where we had our VCF files. I'm going to create a new data table called analysis. And now I've read documentation on PIC-SURE and how to use PIC-SURE from Terra. And I decided that anyone who's going to go through this process needs to have lots of practice. So here in this notebook, I'm copying the current PIC-SURE API 101 notebook from the set of resources provided by BioData Catalyst powered by PIC-SURE. And with this with this notebook, we will be able to examine how the current setup of PIC-SURE as it relates to Terra is. It does change occasionally, and I think it's always very important to basically refresh your knowledge every few months or every 6 months or year, because things do change. So what I've done is I've copied a new notebook to my workspace and localized some files made available in another workspace to my current workspace and copied them to the virtual machine, the cloud environment that's running. I think I can go on to the next. The next bit is I also read in the PIC-SURE documentation that it's necessary to obtain a user-specific security token and save in a file called token.txt. So I'm going to kind of play through it because this is actually a very unique code that shouldn't be shared. So I'm going to pretend that the security token is XXXXX, but for you would replace it by the security token that you copy from the PIC-SURE website. So here I am making a file called token.txt as described in the PIC-SURE documentation. Oh, actually here what I'm doing is I'm examining the files that I had copied over in the previous step. There you go. This is now I've created the token.txt file. And just because I want to make sure that all those X's made it their way to the file correctly, I can do a quick printout of that file. All right. So we're beginning to see how some of the pieces of BioData Catalyst, the multiple parts of the ecosystem are fitting together. We've seen how Gen 3 can do an export of files to a Terra workspace. And now we've seen how we can begin to link a PIC-SURE profile to a current workspace. And the next step we're going to see, probably, you know what, I'm going to not go through this. This is a notebook. I'll just go and stop through a couple places where all the necessary libraries are installed. And this is the notebook that could change over time. So I think that whenever you start a fresh analysis, it's probably a good idea to grab the most recent set of installation steps. And that's one reason why this particular notebook is not going to hang out with my template workspace. It'll be obtained as part of that first step of the template workspace. All right, I'm not going to, the video is available, will be made available later, but let's get into actually preparing files for analysis. So as this particular video plays, I may stop and pause along the way so that we can stop and talk about what we're seeing on the screen. So again, I have just a few libraries to either install or load. And as you can see, I'm going to pause this right here. All right, the first step is, oh, you know what? It needs to go back. I think I scroll back up a bit. Oh no, what do I do? Okay, I copy my PIC-SURE ID. The steps prior to this in this notebook are just to load the set of libraries required. I've already done the installation and the connection to PIC-SURE. I actually might, after this step, I might go back up and show you how I'm connecting to the PIC-SURE resource, or not. Everything that you need is available in this particular file. So even though I didn't stop and show you more about this particular step, now I am. All right, I'm now connecting to the PIC-SURE resource. It's taking a minute. Now I'm waiting. And since I haven't cut any time from any of these videos, we will wait, or maybe we'll skip forward a little bit. All right. So importantly, once you know, well, I know which files I've trying, or what variables I'm trying to obtain. So at this point, work has gone into deciding how I was going to run my analysis and identifying which columns I would need. So what we're looking here is just a printout of the columns available to us from the query that I showed you earlier. You can see demographic columns, blood pressure columns, and then some of these more general TopMed subject, or so study ID, the VCF sample ID, your cohort consent groups, etc. So right now we're just looking at the data file that I've just downloaded from PIC-SURE. So this is the step where I have now imported that existing file with that token that I copied from the PIC-SURE website when I created my query. And I've run this summary. And I'm now interacting with this data frame in R, just like I had read it in from a file. So I know which columns I need to grab. And I've decided to rename the columns into simpler words that are easier to type, because you can see that just by virtue of the way the data is organized and maintaining the provenance of the data, it's important to have these long names from the source. But now that we're doing analysis, we're going to replace it with short names. All right, so I'm running my, I'm manipulating my data frame to make sure I have all the columns I need. And I am, specifying my outcome variable, which I'll use in my genetic association analysis, the covariates I want to use. Now I'm starting to update some of the data models, those tables in my workspace, to be used as input to my workflows. This is one of the powerful features of Terra, is that you can, from your interactive notebook, you can set variables in the workspace data model that later are used as inputs in the workflows. So I am going to create a file called consent grouped table, which will be useful later on. And I'm actually at this step creating files for each cohort consent group that has the BCF sample IDs in that consent group. So I will be using that later on when to filter my BCF file to those individuals in my phenotype file. So I've set up, in this example, 3 different files that have BCF sample IDs that are specific to the various groups. And now here you can see I'm doing a little debugging because what I had expected to see wasn't there. So in my, this particular function lists the content of the Google bucket. And now I'm importing the consent group tables data frame that I just created. And now I am preparing an R data file to use as input to the null model step of my genesis analysis pipeline. And I am putting it in this analysis table in my data model. Okay, I think at this point, all right, there's just a little bit more of my video. So now from not having these tables, you can see how they now appear in my data model. So prior to running the code that we just saw, we didn't have additional, we didn't have the analysis table or consent group, or the consent group table. This is what we just did in this one. We did the analysis table in the previous thing. And then you can see the files that I created. All right, I'm going to stop here. And I'll ask, are there any questions? Maybe we'll take a second to look interactively at our workspace. and take some time looking at these tables. So in that last script, I actually filled in, I created more data for this analysis table. I filled in the covariates that I'll be studying, the outcome, and a link to the phenotype file. So later on, when I run my workflow my analysis workflow, I can specify which row of this table I want to use as to input my outcome variable, my covariates, and the phenotype file. All right, I'll pause and see if there are any questions.
No
All right.
questions in the chat.
What's that?
Oh, I was just saying no questions in the chat.
All right, wonderful. So the next, I have 2 more videos and then I'll do something interactive. So let's go back to the videos. All right, now we have prepared our phenotype data for the analysis. We have to now prepare our genotype data for analysis. And as we had observed, data was organized by cohort consent group. So there were 2 consent groups for the MESA cohort and 1 consent group for the Amish cohort. And the VCF files were separated by this consent group. So there are 3 VCF files for every chromosome. So one of the steps that we need to do in order to prepare the data for the rare variant association analysis is to filter the VCF files by your analysis sample, and then merge the 3 files per chromosome into a single VCF file per chromosome. And then we're going to convert that VCF file to a GDS file, which is the file format for genetic data that's required for the genesis analysis pipeline. So let's get in and see how this first script will allow us to prepare our data model for the view for the subset and merge. So like before, I just have a couple pieces of code in the beginning that just obtain the variables for my analysis. Now I am going to look at the reference file data table from the workspace. And this will have one row for every VCF file that's currently that I exported earlier. I do need to do a bit of manual mapping in order to match the TopMed dbGAP accession ID to the study-specific dbGAP accession ID. But and now I am going to manipulate my reference files table to add a column so that every row that has the appropriate list file of IDs for the VCF file in that row. So there are 3 collections of cohorts, 1 chromosome per, or 1 file per chromosome. So it's 3 times that, 3 times 23 chromosome files. So that was kind of quick, and I didn't quite pause between it. So what I might do is go back to the notebook and just step through this a little more slowly. So this was the part 1. And another nice feature of Terra is the ability to see a preview, a rendered notebook, without having to actually have your cloud environment running. So I'm not in edit mode or interactive analysis mode, but I can see what the output was when I last saved this within an interactive analysis. So like I said, we looked at the reference file. So there's 1 row per chromosome, and each row represents 1 VCF file from 1 chromosome. And my task in this particular notebook is to add another column to this table that has the correct list of IDs, so a file that has a list of IDs. Oh, you don't have to look at all those. So I've set up another table in my workspace called consent group, which has the file of VCF sample IDs for each of the consent groups. And then I do a little bit of manual mapping. There's probably a simpler way to do that, but I hadn't found it over the last few weeks. This could have been rendered a little bit better, so who knows why that is the way it is. But in this set of code, I'm actually, you can see, yes, I have added the VCF samples ID, and now I'm printing it in this line. So, and only pulling out these columns. So now in every row, I have the appropriate sample IDs file. This was necessary to make the workflow step go more smoothly. So the way that the workflow runs, here I'm going to give a demonstration of how we import a workflow from one of two sources. The first source is the Terra workflow repository, and the second source is DocStore. So I actually obtained a workflow from DocStore from the Data BioSphere team and ran it, but noticed that I needed to add a bit of more information. So one of the videos that I have actually shows me going to the Broad methods repository and importing it, but because we only have a few more minutes left of this demo, I'm going to just talk through some features of this particular workflow. And the other video will be available to all of you to watch later. So I always examine the WDL. It's pretty readable once you get used to how it is structured. The workflow has this particular label, has these inputs, and the tasks that are called. And this particular workflow only has a single task. And so in the task, you can see what the commands are, and just like reading a bash script, understand what's happening. And oh, I see here, this BCF tools view command is going to be run. All right, so I'm trying to understand what's happening here. The modification that I needed to make, which is why it's in my own repository, but it's publicly available for all of you, is to add a disks parameter to the workspace runtime environment. So I added this line. This demonstrates how straightforward it is to edit a WDL. For the purposes of this demonstration, I wanted to convert each of the chromosome-specific cohort consent group specific files to a subsetted VCF file that only has those individuals that I want to study from my phenotype data. So I'm going to create a pipe between inputs and outputs using my data model. So I'm going to go take a look at the available data tables in my data model to use as input. And for this, I want to use the reference file. And I could select one or as many of these rows as I wanted to run at a time. Earlier today, when I created this workspace, I chose to run only chromosome 20 or chromosome 21. But you can see I intend to fill in this subsetted VCF file column with output. So if I wanted to select this particular row, which I can see is chromosome 1 from the Amish cohort, I would say, okay, I've selected that row from the reference file data table. As the various inputs, I've specified using the this phrase that I want to use the DERS URI column from the reference file data table as my input VCF. And notice this is the PFB DRS DERS URI. So it's going to do some fancy behind the scenes work to go grab that file from somewhere on the internet, that it's stored securely and I can access it because I have permission. I've set up additional parameters for this example. I do need to use an additional view options, which I saw when I examined the script, the WDL script I could do. And I am using call caching, which allows me in multiple task workflows to basically pick up where I left off if there's a failure on a subsequent task. So this does speed up your analysis quite a bit. I make a note. So this is Amish cohort, chromosome 1, and I can launch the analysis. So now this is actually going to queue and wait. I have to wait a little while. It's going to go off into a virtual machine somewhere that I don't have to think about. The script is going to be run on the input files with the input parameters that I've specified, and I can kind of watch as it runs, and if it fails, that's when we can kind of see the various output here. You can see my comment. You can add tags to things. There's a lot of different ways of organizing your analysis. I'm going to stop here and see if there are any questions. Just to review, I've just run, I ran a notebook that set up my data model, and then I configured my workflow after importing it from the Terra data the Terra workflow repository. I configured the workflow, chose the appropriate entity type from my data model to use, selected the data, and made sure that all the attributes, so basically the workflow inputs were set. And sometimes you have to do it a couple times until you get it right, but you get used to it. All right, it is 54 minutes after the hour, and I think that instead of going through the next part, I'll stop here and we can have a bit of discussion. If people want to, we can spend about 5 minutes generally talking about our experience in Terra. And Yaling, you can go ahead and ask your question. Oh, I think you're muted still. I can't hear you yet. I don't know if there's anything we can do, Tiffany.
I don't think so.
Oh, Yaling, if you want to type your question into the chat, I'll take a look at it. Thanks. These videos and the tutorial and the documentation will all be made available for all of you too.
Yeah, I'll just add, I'm going to add this video and the presentations and everything to a training article in the Terra support knowledge base. So there's a training events section. So just check back there tomorrow or the next day, and you'll see everything should be ready.
All right.
I think I can speak
Yes,
through
we
the
can hear
phone
you
now.
now. Yes. Oh, look at you yelling. That's great. So go ahead and ask your
Yeah, I
question.
think it's better to explain. I'm just wondering about the experience of the PIC-SURE APIs. Just I don't have experiences with that one. I used to download the harmonized phenotypes, and then I just deal with everything in the Jupyter notebook.
All right.
And
Well,
I was wondering about the strength of using these API notebooks. Yeah.
the strength.
Like also like the strengths, like why we use API instead of download files.
Oh, I
itself.
see. From
Yeah.
PIC-SURE.
Yeah, exactly.
All right. So I'm actually going to first show you what public resources are available in Terra to learn more about public repertoire. So there are 2 workspaces that will be helpful to all of us as we learn more about using the various components of BioData Catalyst. So the first workspace, and I'm an R programmer, so I'll go to this workspace that has examples of using PIC-SURE in using R code. And here you can see there's pretty substantial documentation and instructions on setting up your access. So if this does change, you'll know where to come. One place you can go to basically stay up to date. So we've got some nice documentation here talking about that security token step. And there are a number of notebooks that illustrate how to use the PIC-SURE API. I'm not an expert. I do a lot of examining these, demonstration workbooks, notebooks to learn how to do things. And there is this notebook on using harmonized variables, which I used to learn and understand the steps. But this actually you can use to you can learn how to use the API directly to search and create queries. I chose to do it through the PIC-SURE website because it was a little easier for me. But others who are more programmatically or who like to program or want to have all these data available to them through an API would spend, would learn how to interact with the API. So I would encourage everybody to just start using these tutorial notebooks and learn more, and examine the documentation that is available in BioData Catalyst. There are a lot of resources and actually people who are willing to help if there are questions. So there's the resources for obtaining instructions from BioData Catalyst.
Sounds good. Thank you.
Oh, thank you. All right, so it is almost 4 o'clock. And like I said, the additional tutorials with me speaking will be made available in a collection of resources. And I look forward to everyone examining them and giving us feedback as top meta-analyses proceed.
Thank you, Lisa.
You're very welcome.